Coming up next on Futures in Biotech, Dr. Gordon Lithgow bridges the science of protein folding and life extension.
Netcasts you love.
From people you trust.
This is Twit. Bandwidth for Futures in Biotech is provided by Cachefly at cachefly.com.
This is Futures in Biotech, episode 81, recorded June 17th, 2011. Worm research. The buck starts here.
I believe that biotech is the next frontier.
Probably the greatest intellectual revolution that's ever taken place in man's history. DNA is the code for life. We're actually beginning to understand how life works, which I think is just something that's mind-blowing in and of itself.
There was going to be a genetic component to aging.
How long was there the extension?
About 30-40% for humans. That would equate to something like 20 to 30 years.
How close are we to actually having a therapy?
Ballpark, 10 years.
It's potentially one of the things that'll end up rocking the world the same way that people said, oh, the sun's the center of the universe, oh, this and that and everything. And now here's somebody who can come out and say, hey, look, here's how we compare it to our closest evolutionary relative.
So today we are inviting Dr. Gordon Lithgow from the Buck Institute for Research in Aging. And this is the third episode coming from the Buck Institute. And our guest host today is Dr. Simon Melov. And he's been very, very helpful at producing these shows, but also helping guide the conversations. And he's been working with Dr. Lithgow he's known Dr. Lithgow for at least 20 years or close to that. Maybe you could tell us a little bit about his work, him and his work.
Sure. Gordon has been really working on the genetics of aging and lifespan extension in C. elegans since before it was fashionable, which is about 20 years ago. In fact, I postdoced with Gordon in Tom Johnson's lab, who was in general considered to be one of the first investigators to seriously use model systems to investigate the genetics of aging. And Gordon and I attended meetings in the early 90s where people essentially ridiculed the idea that you could study aging at all in genetic systems because it was just too complicated. And so that subsequently proved to be wrong, and Gordon has gone from strength to strength in terms of using C. elegans as a tool to uncover meaningful biological pathways which show real promise in terms of attenuating, altering the trajectory of aging systems. So I think that sort of sums Gordon up. He's got predominantly a focus on stress mechanisms in the worm, and that's illustrated beautifully by this paper he just recently published in Nature.
His work, I think, is a beautiful example of pharmacogenetics, right? Doing early drug discovery and combine it with the power of a fantastic genetic model to really determine mechanism of action of that drug. Right. And it's a paradigm shifting in the field. In the field of health, really, because aging is it's not the aging that kills you; it's the diseases that come along with aging, right?
Right. Although there's something which is really misunderstood and confusing to a lot of people in this area, in the sense that aging is the single biggest risk factor for really the vast majority of all disease. So if we understand aging, we've really got an insight into these diseases which kill us. However, if you eliminate the diseases themselves by some magic pill, then you don't really do much to the natural lifespan. You extend it by a few modest years. It doesn't really do a lot. On the other hand, if you address the underlying cause, the biology of aging, then you get dramatic increases in lifespan. So these two things, diseases of aging and the biology of aging, are inextricably linked. And I think that has only just recently begun to be really understood at a basic level by a number of investigators in the field. It's starting to become a well discussed topic, but the study of aging as the single biggest risk factor for all of these diseases, cancer, heart disease, neurodegenerative disease, stroke, you name it, is becoming more and more appreciated, I think.
Well, the FDA won't allow you to do a test on aging if aging is not a disease. So the whole process is tough.
Yeah, it is. So that's typically why companies focusing on this area pick a disease and say this is our indication, because we can't study aging. And in fact, that's right, because we don't really have good biomarkers for aging. Aging is like art. I mean, you know it when you see it, but it's very difficult to define. The terminology and the definitions which really work are those which focus along a decline in function. But that's so nebulous. I mean, what does that mean? Okay, muscles decline in function with age, but using that as a metric for evaluating whether or not some intervention is effective or not would be fairly limited. So I think that's one of the difficulties, is we can't describe aging, and we're still struggling to some extent in terms of defining what aging is at a systems-wide level. We're getting better at defining what aging is at a single cell level or a tissue-specific level, but still, the definition of what aging is, it eludes us somewhat.
Well, I think after our conversation with Dr. Lithgow, it's going to make a whole lot more sense. I mean, I'm hoping that people can take away from this conversation a real understanding, or not an understanding, a vision of how this will go from the lab to the clinic. And so our guest is Dr. Gordon Lithgow. He's professor at the Buck Institute for Research in Aging. He did his Ph.D. in genetics at the University of Glasgow in Scotland. His postdoctoral training at the Institute of Behavioral Genetics at the University of Colorado in Boulder. He's was a senior lecturer in molecular gerontology at the School of Biological Sciences at the University of Manchester in England before moving to the Buck Institute in 2001. He's principal investigator and director of the Buck Institute's Interdisciplinary Research Consortium on Geroscience. He's also principal investigator of the Larry L. Hillblom Network on Chemical Biology of Aging, and is the coordinator of the Hillblom Center for Biology of Aging Support Award. So I welcome him to the show.
Thank you. It's a pleasure to be here.
I'd like to sort of preface this a little bit. I mean, this is not a show that's going to be out of context, and this is a field we've touched on a little bit, even starting 5 years ago. Almost to the day, we interviewed Dr. Leonard Garanti, who discovered the sirtuins with the president of the institute you're at
Buck.
The Buck Institute for, the name keeps changing, but the Buck Institute for Research in Aging.
That's right.
And both are fantastic, contributors to the field. We've interviewed Aubrey de Grey, who sort of comes in from a completely different angle to science, which was very, very unique to his own work. We've interviewed Cynthia Kenyon, and Cynthia Kenyon's work was using some of the similar models that you use. So
That's right.
If people want to hear a little bit more about C. elegans, they can listen to Cynthia Kenyon's episode, which is episode 36. And they can also listen to 2 episodes with Marty Chalfie, who won, I think, the 2007 Nobel Prize for his work on GFP, and did it in C. elegans as well. Then we had an episode with Dr. Kennedy, who was Dr. Garanti's student and the president of your institute, and then Judith Campisi, who was fantastic, who's a colleague of yours. And so people can go back to those episodes. That's 2, 30, 36, 73, and 74. But the context of your work and the contribution is remarkable in itself, and brings a whole new angle to the research in aging. But before we get on to so much the implications of it, could we talk a little bit about your recent study that was published in Nature, and some of the techniques you used, and sort of how that science was initiated, how it started, and maybe you could talk us a little bit through through the findings of that study. It'd be from a technical perspective. I want to get to the nitty-gritty. I want to know what it's like to be in your lab.
Yeah, okay. Let me just 2 words on the context. The first thing is that we've been interested in finding chemical compounds that extend lifespan for a long time now. And we've also been interested in the notion that protein conformation, protein shape, is really important in aging. And the recent paper in Nature was the brainchild of a postdoc in my lab, Silvestre Alves. And so he was aware that we were working with compounds and doing chemical screens, and he was also aware of this notion of protein shape or protein conformation being important in aging. And actually, he went back to a concept that's over 100 years old from Paul Ehrlich, who you might remember from your microbiology lectures. Paul was the guy, the German scientist who was studying chemical dyes and was interested in dyes that would stick to particular components of cells, particularly pathogens. And in doing so, he simply thought if these things can stick to pathogens, then they may do something, they may be harmful to the pathogen and may therefore be a good way to get rid of them. And so this was the magic bullet concept. And that concept is what Silvestre did in the lab. My lab is kind of informal, and people can go off and do some of their own things and their own ideas and follow their own experiments. And this was one of those experiments that Silvestre did that I didn't know about for a while. But he took some dyes that were known to bind not bacteria, but components of age-related disease. These dyes are used in histochemical staining to visualize amyloid. And of course, amyloid is the stuff that accumulates in Alzheimer's brains and other neurological diseases. And Silvestre was aware of these dyes, having been trained as a neurochemist himself. And so he started to apply these dyes to age-related disease conditions. So let me explain the model here. The model is C. elegans. It's the worm. And the worm has been engineered by a couple of people, Chris Link and Rick Morimoto and some others, to exhibit aspects of neurological disease. So you can express neurotoxic proteins like beta amyloid, the Huntington protein, and so on. And what happens in the worms is that these tend to form aggregates, which you can visualize. These are large aggregates in the animal. And then the animals get sick, they become paralyzed. So actually, it's quite easy to look for genetic modifiers, or in this case, chemical compounds, that change the course of that whole pathology, that change the course of a healthy worm to a paralyzed worm.
Let me just stop you just for a sec. So to get a little recap here, this beta amyloid protein, for those that are not familiar with it, when the protein misfolds, it forms a polymer, and that polymer forms these nanotubes, and those accumulate in the brain of patients with Alzheimer's?
Yes, that's correct. There's a whole cascade of events from a normal A-beta protein, although we don't really know what the normal function of A-beta is, but there's a cascade of events all the way down to large insoluble aggregates. And these are constituents of plaques, for example, in the Alzheimer's brain. So it's that aspect of Alzheimer's pathology that Chris Link was able to engineer into C. elegans. You remember C. elegans? I've got a model here. So
yeah, show it. That's really fantastic.
Sure. The C. elegans is a millimeter in size normally, and the thing that you can see here is that the animal is transparent. You can see all the tissues, right? So here's some fertilized embryos here. Oh, sorry. Here, for example, here. Unfertilized oocytes. Here's the embryos here, actually. And so you can visualize aggregates in these animals quite easily. So if you express A-beta in the muscle, for example, you see big, really big bodies of A-beta amyloid accumulating in the cells of the muscle. And this,
by the way, those are the size of a comma. We do have about maybe a half-second delay here. But so they're the size of a comma, and actually they sometimes curve like a comma.
That's right.
And they swim around in a Petri dish.
They do. They swim around on an agar surface.
And they eat yeast cells?
And they eat actually E. coli in the lab. So they eat bacteria, yeah. So actually what you do then is to take an agar plate of these things and pipette on a solution of a particular chemical. So Silvestre was looking at these dyes that are known to bind amyloid, and he would pipette some of the dye onto the plate, let it go in, add the worms, and then let the worms age. Now the worms age in about 20 days. You got a 20-day lifespan. And you can really see the normal aging progression of the worm, partly because it's transparent. You can look down, and Simon will tell you there's all sorts of interesting things going on in the worm inside as it ages. One of the things in
these
these
are
worms,
mutated, though, right? To have that disease, or to have the
correct.
expression of the protein that aggregates.
That's correct. Actually, in the paper, we've got all sorts of different kinds of worms. We've got those that have been engineered to mimic aspects of Alzheimer's and Huntington's disease. And we've also got normal worms that are not engineered in any way at all.
Okay.
So let's take the disease worms. What happens with the disease worms is that some of these amyloid binding dyes postpone the onset of the formation of the aggregates. And when that happens, the worms seem healthier for longer. So you don't see this paralysis kicking in. So the compounds seem to slow down the progression of this disease pathology. Now, if you take normal worms that don't get the disease, they don't have beta amyloid, and you squirt the compound on those and let them grow and let them age in the presence of this compound over 20 days, you see a lifespan extension.
Why?
So on one hand, you've slowed down an aging disease pathology, but then just as exciting, you seem to have slowed down aging itself. And these animals live anywhere from 30 to 70% longer in the presence of the dyes.
And that's first of all, let me just, it's Silvestri Alavarez?
Alaves, A-L-A-V-E-Z.
Alaves, sorry, yes.
Yeah.
Silvestri Alaves. Brilliant idea.
Great idea.
It's one of those genius ideas that you take to a lab and holy smokes if it works.
Well, it's also one of those ideas that if you actually sat down and carefully thought about that experiment, you'd probably come to the conclusion that it wasn't going to work, and maybe it wasn't a particularly good idea anyway, and you wouldn't do it. And that's the great thing about it's a great thing about having that freedom. You just do the experiment, see what happens, and in this case, it turns out to be a brilliant idea.
Fantastic. But this yellow, it's yellow dye, right? It's a common yellow food coloring dye.
It's not really used in foods. It's used in histochemical staining. It's called basic yellow 1.
Basic yellow 1.
Or thioflavin T. That's right.
All right. So it binds. This is one dye, though, that people use to identify the misfolded proteins that form these fibers specifically, right? It's a traditional dye for amyloid pathology.
Absolutely, and actually a very, very similar chemical structure is used to visualize amyloid in the brains of living people. This is the famous Pittsburgh compound B. So if this is a labeled compound that can be detected on a PET scan and is actually now being quite commonly used to visualize the amyloid load in people as they age. So it was exciting for us that this is a compound that's already been used for a separate purpose, albeit in people.
Safe. So it's a safe compound as well.
Oh, yeah. I think that the way that we're doing these experiments is that these animals, these worms, are growing in this stuff all the way through their lives. So we can't really say anything about that kind of protocol in humans. The protocol that it's approved for is a very short-term injection of the compound for visualization. It's cleaned out quite quickly. And it is important to say that at high concentrations, it's toxic in worms, probably just like most chemicals.
Okay, so you looked at the thioflavin T compound that binds amyloids, and you tested it in the worms. You monitored the worms over 40 days?
Yeah, 20 days, 20 to 25 days, yeah.
20, 25 days. You have some data points where the worms actually lived really long.
Yeah, no, that's true. Individual worms. That's right. Individual worms can really live out there, up to 70% longer. You can get individuals that will live out to 40 days. That's right.
And there was a specific concentration, 50 micromolar of thioflavin T. You tried up to 500 micromolar. That seemed to kill them. So you go too far and you kill them. And then 1 micromolar, you don't see much of an effect. 10, a little bit of an effect. And the sweet spot is 50 micromolar.
Yeah, that's right. Yeah, nice dose response.
Now, I'm looking at the same figure. So when we're looking at figure 1, and the paper's titled Amyloid-Binding Compounds Maintain Protein Homeostasis During Aging and Extend Lifespan. And I'm going to put a link in the futuresinbiotech.com website to be able to access this paper. I think it's a free paper on PubMed. But if you go to figure 1, you have these nice curves. I can even show. Maybe you can't. The contrast is too high. But so you're looking at percentage of survival over time, and the longer the worms live, there you get a higher percentage that live and then the curve drops off at like 30 days instead of 20, which are the normal worms. But you have two other compounds on here. One that is amino phenyl benzimidazole, BM, or hydroxyphenyl benzothiazole, which, if you look at those dose response curves, right, you're going from, instead of going from 1 micromolar to 500, where the sweet spot's 50, you're going from 1 nanomolar to 1 micromolar. And if I look at what you call HBT or BM, they're working as effectively with I guess you could have gone to a femtomolar, but nanomolar, 1 nanomolar was like in the middle of the sweet spot based on this figure. Maybe I'm not interpreting correctly. But if then you compare that to the thioflavin T, you're looking at a pharmacological effect that's 50,000 times more potent.
That's right.
And when you read the paper, which we'll get to the point of the thioflavin T and the protein homeostasis, I'm looking at this figure and I'm jumping for like joy, because here's a molecule that seems structured. You put it in here because it says similar structural features to THT, but with different pharmacological properties.
That's right.
I then went into PubChem and looked for these compounds and did not find any significant pharmacological properties, but maybe I didn't do my research that well. But BM and HBT are at the 1 nanomolar range, providing a significant extension of lifespan, and they're 50,000 times more potent.
Yes, I'll tell you, I was jumping up and down as well. Okay,
yes,
these
I can imagine.
these are really terrific. They actually, all 3 compounds were developed by a group in Barcelona, and they're actually, we don't talk about this in the paper. You don't have space for this kind of speculation.
No.
But the compounds. But here it is. The compounds are actually hybrids between thioflavin T and a compound called cleoquinol. Now, cleoquinol, some of your listeners will know this. This is a compound that is known to bind metals. It doesn't bind very tightly, but it binds metals, and it's been used in some clinical trials for Alzheimer's disease. And compounds like cleoquinol are being developed as therapeutics because of the metal binding properties. And this group in Barcelona thought, well, wouldn't it be great if you had thioflavin T, which is known to bind amyloid, and you had cleoquinol, which was known to bind metals. Now, the thing that's missing here is that metals are key for amyloid formation. If you had a compound that did both those things, maybe it'd be really effective as a therapeutic. And they did the biochemistry on this. They showed that these hybrid molecules, the HBT and the BM and so on, they do indeed bind metals, and they do indeed slow down the formation of amyloid in vitro. So when Silvestri saw these compounds, he thought, well, this is great. They actually are more drug-like. They appear more drug-like following Lipinski's rules, and they have this additional property. So, as I say, we didn't speculate in the paper, but now we're wondering, are these compounds working at such the effective dose at such a lower concentration because actually they're doing something in addition to the action on amyloid formation? Are they actually also perhaps binding metals?
My thought would be that they're doing exactly what you speculate, but I mean that's the first thing to try. But at 1 nanomolar, let me just suggest that the potency here, while the efficacy isn't 100%, it's only maybe a 30%, 40% improvement in the lifespan. It's not an efficacy issue, it's a potency issue. And that the potency is so strong that you're probably hitting a handful of key proteins whose stabilization leads to life extension. That it's a potency, the potency is so good that your specificity of ligand protein interaction is so strong, you're pharmacologically ready to go. And go ahead.
No, that's a really interesting point. And actually, we don't know what the targets of these compounds are. In a normal worm, for example, there's no amyloid, beta amyloid there, but we suspect that normal worm proteins are forming amyloid-like structures as well. And that may be a few proteins, as you suggest. It may be hundreds, it may be thousands of proteins that can form toxic intermediates on the road to amyloid formation that these compounds are perhaps interacting with and slowing down their formation. So yeah, that's an interesting idea, though, that the potency is revealing that there's a fewer number of targets than what we see with thioflavin T.
But that's what you want. I mean, that's the end point of a drug discovery program or drug development program, is to have that high potency, then go after the structure-activity relationship and then nail it even more. But it's so, you've got this, these compounds are so potent. But this is kind of an Easter egg in the paper, because you don't discuss much of it. You just present it as related compounds that have, but it is beautiful. So I saw that figure as I jumped out, oh my God, 50,000 times more potent than the paper, the compound you're talking about. And then you do a beautiful, dissection of the function of the thioflavin T. So perhaps we could get back to the, I'm hoping that that you, before the publication, that the intellectual property was protected on BM and HBT for development, because you have to leverage that IP to raise the money to get into clinical trials.
Yes, and actually there is some intellectual property around thioflavin T structures, because they have this history of being used as agents to visualize beta amyloid. So we had to pick our way through that, but we did indeed file a preliminary application around the notion that these compounds could be useful in combating age-related disease. So absolutely, we're very interested in developing, going into this space and seeing what's there for therapeutics. And the first stage for us is to start looking at these compounds in more complex animal models, the mouse, of course, and look at neurological disease, but perhaps other diseases where amyloid is a feature as well.
Could I jump in here?
Absolutely.
So I was just going to ask Gordon. Gordon has actually been leading an effort here at the Buck Institute to use the worm as a screening system for identifying agents which may prolong lifespan and prevent age-related disease. And this is the culmination of some of that effort. But Gordon, I was wondering if you could talk a little bit more generally about the strategy of screening compounds and frame that in a historical context, in the sense that, we've been doing research on aging now for more than 20 years in these invertebrate systems. And here we are, we're now identifying compounds really, I would say, sort of almost routinely. Perhaps that's a bit of a stretch, but I think it's certainly not unusual now to identify compounds which extend lifespan in invertebrate systems. And how can we translate this? How can we make this go to the next step, which of course is moving into mammalian systems and ultimately into human beings? Can you give us some of your thoughts on that?
Yeah, well, okay. So you say 20 years, and 20 years is round about the time that we've been doing genetics of aging in invertebrates. Those first mutations from Tom Johnson's lab and Cynthia Kenyon's lab coming out in 1988 and 1993 and so on. And we've learned an awful lot about the genetic architecture of longevity in this time, mainly in invertebrate models, but now also moving into rodents. And what we know is that there's a plethora of potential targets. There are hundreds and hundreds of genes in C. elegans that depress lifespan, and when you knock down the expression of those genes, you see large lifespan extensions. And if each of those is potentially a pharmaceutical target, we're seeing all sorts of different areas. We're seeing endocrine functions, metabolic functions, intracellular signaling pathways. And so you're right, as of 10 years ago, we were all thinking, well, let's get after small molecules that potentially hit these different pathways and extend lifespan. If aging is a common cause for age-related disease, Alzheimer's, Parkinson's, adult cancers, and so on, then possibly this is a really good space to develop therapeutics, because not only are you going after the very earliest events in those age-related diseases, you've actually got a good chance of hitting a number of disease conditions and preventing them. So yeah, the general thinking has been, let's find these compounds that hit these pathways. And of course, we've gone back to the invertebrates because that's where the genes were found in the first place, and they're quite easy to screen large numbers of compounds. C. elegans itself is very amenable to high throughput screening, and a number of labs have been doing this activity. And as you say, it's fairly routine now for someone to come into a lab and find a small molecule that has an effect on lifespan. The difficulty, I guess, is making sure or finding out some sort of mechanistic level of what you've actually done when you do that. And that really will then help in the translation. 20 years of genetics, I don't think we've really helped anyone yet, to be honest. We can't point to a therapeutic that's really been developed through this work. The investment in this area has been fairly modest up until now, but I think we're at a point where we have to decide if really we want to start going down the road toward preventing age-related disease And I think this is a great way to go about it. Find compounds in simple animals that extend lifespan, interfere with aging mechanisms, and then get them into rodent models of age-related disease and see what we have. I think it's a very, very bright future in this area.
Well, you know, for what? How many thousand years? 5,000 years of the history of humankind, we've been going after molecules, or not going after the desire, the fountain of youth, right? Back from down to Cleopatra. So now you've got 15 years. You've been working on it. It's, you know, you're trying to push back, extend health or delay aging. To delay the onset of age-related diseases, which are major ailments to humanity. But, you know, it's okay. You know, 15 years is even 20. Don't worry about it. It's fantastic work. And it's going to take some time before this translates.
Well, the closer you get to 50 years old, the more impatient you get, I guess, that something's going to happen.
Well, the genome was just completed, and, you know, in the scope of things, the proteome hasn't been completed yet. They're only starting the one of the chromosomes for the proteomics of a single chromosome. Hopefully, is the first trial for the human proteome.
Right? Well, I mean, coming back to the question of the emphasis and the focus on this as a potential avenue for reducing the incidence of age-related disease and extending the health span of human beings, it is sort of philosophically interesting that the discussion in the blogosphere and the political sphere in general is on healthcare and you know, how we can improve healthcare, but none of that discussion focuses on increasing basic funding for research which will cure and prevent these diseases. It's all focused on cost cutting and, you know, more beds and better management of the healthcare systems. Seem completely absent from the dialogue is the idea that the real solution to these problems is in more research.
Can I pick that up, Simon? I mean, essentially, until now, what we've had is a science project. I came into this field because I thought the biology might be cool. I thought we might learn some really interesting things by studying aging. I had no concept 20 years ago that we'd be sitting discussing this as a potential way to develop therapeutics and head off a healthcare crisis. I mean, this is something that's really developed over the last 10 years, the understanding that aging mechanisms and disease mechanisms are really tightly coupled, and that therefore there's an opportunity to go in after aging itself as a therapeutic target. So it's true that there's no conversation out there about investment in this particular way to cut through the healthcare crisis, cut through the financial horrible financial implications of what's happening demographically. But here we are, a couple of worm, geneticists actually discussing these issues, and it's because it's now, the first baby boomer turned 65 this year. We're faced with the crisis now.
Well, the threat, the economic threat of baby boomers reaching an age where the onset of diabetes, Alzheimer's, and cancer really weigh on the American economy. There's the precedent for one might just say market-driven science, where there's going to be a value to these compounds getting to the clinic, should create a private gold rush with respect to drug development. And to be positive about it, one must look at these opportunities and look at your work here. And when I see BM, and if you guys have the proper, IP strategy at the Buck Institute for BM and HBT and related compounds, I see this as a huge opportunity, and maybe we should be discussing that in private and contact each other after the show. But I don't. There's so much content in here, and the fun stuff about the fun element about this is such a human condition to age, and not just delaying the aging, but improving health throughout a person's age into their 90s, and living to their genetic potential is an incredible quest, and we always tend to short circuit the discussion away from the technical aspects and get into the philosophical and economic implications of age research in aging. And I'd like to step us back just for a few minutes to get back onto the paper and sort of while these related compounds are extremely potent pharmacologically, at least in the worm, the whole concept of why thioflavin T is creating a significant improvement is not entirely clear from our discussion. It'd be really good if we could get a little bit back into the technical element here.
Sure.
And talk about protein homeostasis. So how does thioflavin T, how did you guys dissect the role of thioflavin T on protein homeostasis and its effect on aging?
Okay, so yeah, no, that's a great question. Really, this was being done in an atmosphere of the last 10 years of genetics, which has strongly suggested to us and to others that protein homeostasis, in other words, the way in which the cell looks after protein shape and maintains protein shape and gets rid of either modified or damaged proteins or misfolded proteins. All that machinery, molecular chaperones and autophagy functions and protein degradation machinery, all of that seems to be important for lifespan determination, as shown by various mutations in C. elegans. So we had this very good idea, beautiful work from Rick Morimoto's lab and Cynthia Kenyon's lab and Chris Link's and others, suggesting that protein homeostasis was a major determinant of longevity. That's kind of exciting because this is, of course, an area where the neuroscientists have been following in relation to Alzheimer's and Huntington's and Parkinson's for quite a while. The notion that neurotoxic forms of proteins, certain conformational forms of proteins, are contributors to those diseases. So there you have aging and age-related disease overlapping in some way. And way back in the early 2000s, we were showing that if you took animals and engineered them to make more molecular chaperones, those proteins that help maintain the shape of other cellular proteins, you could extend the lifespan. So the thinking was that are there chemical equivalents, are there chemical chaperones that would maintain protein conformation? And so Silvestri's idea that a chemical that would bind to amyloid may in some way shake up those protein misfolding pathways, I think is true. Now what we've done is to combine compound treatment with genetic intervention. So we can knock down certain genes like molecular chaperones or autophagy functions and ask if the compounds are still working. And in many cases, when you knock down a protein homeostatic function, the compounds no longer are able to prevent the formation of amyloid quite as well. So the compounds require normal cellular functions. They require protein homeostatic networks to function.
So you basically, the cells have biological pathways, a series of signaling pathways, dip switches in the cell that maintain normal function. And with C. elegans being such a powerful genetic tool like the modern fruit fly, you can go in and selectively block each one of those biological mechanisms to determine how your drug is working, which mechanism, which pathway is it acting through, or maybe it's an indirect result. You knock out that pathway, your drug no longer works. Is that what you mean?
That's the basic premise, yeah. This is pharmacogenetics, right? You combine a pharmacological agent with a genetic manipulation, and you ask if that manipulation is now blocking the action of your agent. You may have hit the target, but more likely you've just hit a process that's required for the compound to work. So in this case, we're seeing that it's not just molecular chaperones and some of the normal protein homeostatic functions. It's also the regulators of those functions. So for example, transcription factors like SKIN1 and heat shock factor 1. These are transcription factors that respond to stress and turn on suites of genes that are required to counteract that stress and let the animal survive. Well, thioflavin T needs those transcription factors for its function. So we can see a twofold thing here mechanistically. One, we suspect that the compounds are interacting with amyloid or misfolding pathways, and that has some sort of effect. And then secondly, we get this second effect where the animals turn on suites of genes that are then able to encode the proteins that then can clean up the system. So we suspect that you need both the presence of the compound and an active protein homeostatic network in order to get the maximum effect.
It's amazing. Well, could that protein homeostasis pathway be required for the response to the toxicity of the drug and then elicit like an immune response that protects the animal?
Yeah, we're wondering about that as well. You know, that there's an old concept that a little bit of stress is actually good for you, hormesis. And actually, way back in the mid-90s, we showed that if you subjected worms to a very small amount of stress, you could actually see a lifespan extension. And we've always been on the lookout for that with compounds. Are compounds really just working through hormesis? Are they stressing the animal responds to the stress, and then they derive benefit during normal aging? This doesn't look quite like the hormesis that we've seen in the past. There's something more selective about the genes that are turned on here. But the principle's correct, you know, that a response of an animal to a drug could in itself be beneficial. And so some of the factors that are being increased by the drug, the animal might be deriving benefit from what is essentially a sort of subtoxic effect.
I'm speechless. This is amazing. So C. elegans is really the tool, and you when you screen with these, you do videotaping of the worms, or do you actually have a student or a graduate student technician monitor their behavior?
Yeah, actually, you know, many people are developing automated ways of looking at the worm and tracking the worm, tracking its movement. We've got an automated assay for worm death, where it takes up a fluorescent dye when it undergoes death. But you know, there's nothing beats a pair of human eyes looking down a microscope. They're able to capture so much more information. We do a lot of our screening by eye. People come in the morning, they get their worm plates out of the incubator, they sit down at a scope for a few hours, and they go in, and occasionally what they're doing is they're going in and touching the animals with a platinum wire to see if they're going to have, you know, respond to that touch. In other assays, we're hitting them with a small amount of fluorescent light, which induces movement, and so the animals that are healthier tend to move faster. But you know, people looking down microscopes is still incredibly valuable to us.
Worm pokers.
Absolutely. Worm wranglers.
How many worms can you screen? How many drugs can you screen? I guess each plate is going to be a different compound.
Oh yeah, I mean, we've in the lab over the last couple of years, we've looked at 25,000 or so, and we go back. We're currently screening a smaller focused library set of natural products. That's obviously a nice way to go if you're thinking about translation. And so we're bringing, you know, different assays to these chemical screens, and actually we're trying to find what compounds come out as hits in different types of assays, because they may be the winners. They may be the ones that are affecting all sorts of different and interesting biology.
Are these isolated natural compounds, or are they extracts?
Yes, they are, and people are looking at extracts as well. There's some really nice published work from Kathy Wolko's lab, for example, showing that blueberry extracts extend worm lifespan. So there's something in the blueberries that's beneficial. Yeah.
I only have one fear about extracts. Is one, enough compound to follow up for the research for the clinical development? Two, synthesis. I mean, you've got sometimes nothing beats a plant at making, plant secondary metabolism is so fantastic that the compounds might require 100 steps of synthesis. And if your compound, the most effective compound you have is requires 100 steps, then you basically have to build a farm to purify that compound. And then you might just end up with a dead end, even though it's so effective. But the biology of a plant pharmaceutical, I mean, they're so potent. Nature made them that way, right?
Yeah, and you know, the thinking there is that also that the worms have been exposed to plant compounds for, you know, many hundreds of millions of years, and so you've got this notion that maybe they already have a connection with natural products that maybe they don't have with some synthetic compounds. You know, it's an idea that comes out of the notion that there appears to be more hits come out of natural product libraries sometimes than the diversity sets. Although we've screened diversity libraries as well, and we do find things there that extend lifespan very well indeed. So we're following all these pursuits. You know, I mean, I think that this is we love this paper. We love this story that these amyloid binding dyes are doing something very potent, and it's nice that we think we've got a handle on a mechanism. These might not be the ones, though. It may be that we need to focus on other pathways and other mechanisms. In the end, perhaps what we're looking at is combination therapies, where we look at protein conformation and protein homeostasis, but we're also protecting against DNA damage or oxidative stress and so on. So I think at all points, again, I'm sorry, you get a couple of scientists together, they start talking about funding, but it really, it points to the need for investment in this area right now, that lots of labs, including ourselves, have all these interesting compounds, and we need to pursue many of them all at the same time to really, come out with the winners here.
Well,
Go ahead.
I
was just going to say, in terms of the funding again, I think the proportion of the NIH budget which has been spent on the biology of aging research is minuscule over a very long period of time. It's very, very tiny. Gordon, I think you have a number. Yeah.
Oh, I estimated it was 0.1% of the total NIH budget is devoted to understanding the biology of aging, and that excludes the expenditures on Alzheimer's, more traditional approaches to Alzheimer's disease and cancer and so on. But this area of thinking about the biology of aging as the major risk factor for human disease and studying that is quite a small investment. And you can think, okay, 20 years ago, some guys playing around with worms and flies. Well, that's what you'd expect. I think the science is incredibly powerful now. It points to the fact that we can do something much more significant in human health than I ever imagined, and I hope the funding situation is going to change.
Your work has really allowed sort of going from a more, frontier. And funding science when it's really, really, really frontier, people, that you pay and the success is very low at frontier end when you're at the bleeding edge. It's high risk, high reward potentially. But you've seen your work, this work here, and the work of the past few years on protein folding and compounds that modulate protein folding and their role in aging has sort of pushed back that frontier to a point where now it seems like a very plausible R01 for the National Institute for Neurological Disorders and Stroke, one of the big, highest funded branches of the NIH. So I think there's a lot of promise. I think there's more funding ahead of you than behind you.
That's nice to hear. Can we put you down as a reviewer, Marc? That'd be great.
Well, I have reviewed you, and we're going to put this online, and people are going to hear it. Your work was selected for presentation here on Futures in Biotech, which is an important element towards a Nobel Prize. So for all out there who are on that track, please come and visit us on Twit. One last question I'd like to ask you, is... Eric Kandel, who's done a tremendous amount of work on memory and
Yes.
molecular mechanisms of memory, and he's been on the show. People want to go back and just Google Eric Kandel, Futures in Biotech. He talks a little bit about it, but in 2009, when I went to the neuroscience meeting in 2009, I didn't go last year, but he presented, he was a keynote speaker, and he presented work where the CPEB, I think is the name of the protein, which is an amyloid-like protein, which is involved in long-term memory potentiation. And here I'm thinking neurological disorders, if you're looking at compounds that bind polymer-shaped, structures in the brain which are pathogenic, could you potentially interfere with the proper formation of these non-pathogenic fibers involved in long-term memory potentiation? Not that I worry about it. I mean, I'd still take the drug if I was having amyloid plaques forming, but what are your thoughts on long-term memory potentiation, Kandel's work, and protein folding? It's hard to frame these questions, but
it's a terrific question, and actually, this has been pointed out to us in a number of ways. People have pointed out that the kinds of concentrations of agents that would slow down aggregate formation in the human brain would be really high for a drug. This wouldn't be a catalytic process. This would be in some way, almost for every polymer, you need to have a certain load of compound, and so in many ways, this general notion of preventing fibril formation has been poo-pooed by the neuroscience community. When we take all of that on board, I mean, I think that's all true, and also this notion that there may be natural processes that involve certain type of fibril processes that you don't want to interfere with, of course, and just like any drug, you've got to minimize side effects any way you can by structure activity and structure function studies. I think that all of that's true, and we have to take all of that on board, but we don't really know what's happening with these compounds yet. The compounds that we've worked on, as I say, it seems to be a two-stage process. There seems to perhaps be a direct interaction, but also this is causing the cell to respond differently. The cells are turning on other genes, and things are being cleaned up at a faster rate. I think that we're just at the outset here of trying to explain how compounds like this have biological effects, and there may be some surprises, and I hope what happens is that we'll find out that we can see a space where we can minimize age-related protein aggregation, protein misfolding. We can minimize that. We can induce cellular endogenous processes that are best suited to cope with that. And we know they cope with it very well in young animals. And in this way, we can postpone the onset of age-related disease.
That's amazing. Are you going to, this is, are you going to focus now on, for the next few years, on the pharmacogenetics and early stage drug discovery? Or are you going to take a few of these compounds and drive them through towards small mammalian models?
Yes, and yes. I think that we're an academic lab. We're still doing science projects, and we'll continue to. That will probably be my major focus. But I'm really excited by all of this. I'm really excited by the potential that the worm community and the fly community could make a big contribution here. So I think we want to convince our colleagues working on age-related disease models that this could be an interesting area that they should look at. We want to begin initiating conversations with clinicians, some of whom might be sympathetic to thinking about how some of these compounds that already have been in humans, maybe we can think of endpoints for clinical trials. And in the end, we also want to have the lab generating the newer compounds based on new screens that actually might be the winners in the end. So the Buck Institute is all about basic research, but basic research that can be translated into meaningful therapies or preventions. So I hold my hand up and say, yeah, let's do it all.
You're going to help probably 1 billion people.
If I could help one or two, that would be fantastic.
At least
we've helped a few million worms so far.
1 billion per generation. Oh, absolutely. The worms are happy. You're looking at 1 billion people per generation. So I thank you for your work, and I congratulate you on this paper, and congratulations to Dr. Alaves as well.
Well, thank you very much indeed for your interest.
Ventapilli, Zucker,
yes,
and Plang as well.
That's right. Yeah,
fantastic.
All made fantastic contributions.
Well, keep it up, and we should get you on, and we should have a conversation afterwards too, and maybe talk to your tech transfer office.
Right, you bet. I'll be on the phone to them right now.
I'm moving to Nevada. Well, it's really convenient because the Twit Cottage is just 10 miles from you guys. It's
fantastic.
My goal in life was to have a podcast and a company.
You could get this check signed this afternoon. In no doubt in my mind is this going to translate. This your work is beautiful traditional science, and it's moving, but it's moving the frontier forward. So the Buck Institute's doing such a great job, and I appreciate the help of Simon on bringing these shows to the forefront of futures in biotech, and to Chris Ribelot as well for bringing this to our attention. So thank you. Gordon, for coming on the show. Thank you, Mark. Thanks for your interest.
Speak to you soon.
Bye-bye now.
First, I'd like to thank Dr. Simon Melov, who's an associate professor at the Buck Institute for Research in Aging. Thank you for coming on the show and helping produce all these great shows on aging, Simon.
No problem. Thanks very much.
It's awesome. I'd also like to thank our guest, Dr. Gordon Lithgow. He's also professor at the Buck Institute for Research in Aging in Novato, California. As well, John Slanina. Yeah, he did all our production over at the cottage today. So thank you for handling the audio and video boards. I'd also like to thank the team that make this possible. Leo Laporte, Lisa Cancel, Frederic Louis, Eileen Rivera, Tony Wang, Mike Taylor, Bert McQuinn, Jeff Stewart, Jason Howell, and of course, Dicky Bartolo. I like to throw his name in there, even though he doesn't do anything. I'm just a huge fan of his on the Twit network. Lastly, I'd like to thank Philip Pelletier and Will Hall for the opening and closing themes. If you have any comments or suggestions, you can contact me at Marc, M-A-R-C, at twit.tv, or on Twitter at Marc Pelletier. For Futures in Biotech, I'm Marc Pelletier.